So we have a very different MO than most people. So it's a little bit difficult to wrap the minds around because we're not angling for anything except to help you. That makes me really happy. That's why I'm a public servant. I really love this work because I get to help people, and that's my job, is to help people out. I learn things all the time. I'm a lifelong learner. I'm out hunting bees all the time. I then get to distill this and put this into practice in a public facing way. I can't really go like, look, we won all these awards, or great, but there's a lot of the mycelia. There's a lot of mycelia, not a lot of mushrooms. How about that?
From the Oregon State University Extension Service, this is Pollination, a podcast that tells the stories of researchers, land managers, and people like you who are making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melathopoulos, Associate Professor of Pollinator Health in the Department of Horticulture. I am so excited this week, more excited than usual. I'm finally catching up with Sam Droege. Now, I know a lot of you know Sam. He's with the U.S. Geological Survey, and a lot of you know the person he works closely with, Clare Maffei, who's with the U.S. Fish and Wildlife Service. And we're going to talk to them about the origin and evolution of this really great, mighty bee monitoring and inventory lab that they work at. Many of you know it's an important hub for bee biodiversity across the U.S., but you may not know where it came from. So in this podcast, we learn, I didn't know this, it started off as a lab that was dedicated to monitoring birds, and then it transitioned into bees. And Sam recalls these early challenges. There's birds, you can identify them by song and looking at them, but bees, they vary from year to year, identification bottlenecks. It's really fascinating because we hear about how they leverage all of these challenges into these new things that we all come to depend on. New sampling techniques, the very handy bee manual, the Discover Life online ID guides, the photographic resources, all of this came from confronting these challenges. Now, Clare outlines her path in the podcast. She started with the U.S. Fish and Wildlife Service Pollinator Fellowship, and she became a position that provided support to our national refuges, producing updated inventory and monitoring protocols, data management, and just recently published over 500,000 records of bee occurrences to GBIF. Together, they really encapsulate this spirit of the lab of working with everybody, students, refuges, parks, researchers, and kind of pulling together this network even amidst this uncertain funding situation that's been going on for a couple of years. It's a great episode with some of the hardest working and generous scientists in the field. So let's join Sam and Clare for an extended podcast this week on pollination. All right. Well, we're rolling. I'm so delighted to have you both on pollination. Welcome, Clare, and welcome, Sam.
Thank you.
Hello.
It's good to be here. I'm looking forward to the conversations.
I think I've had you on the show before, Sam. I don't know. We had you in Corvallis. We've had you both in Corvallis before.
Right. I mean, you were teaching a class, if I want to say, very early on in the Oregon Bee Atlas, and you said, we should talk to Sam about monitoring techniques or catching techniques. And I thought it was just, I'm talking to the class, but it turned into a podcast somewhere.
That's how I usually, that's how I roll. Yeah.
Yeah. Okay.
Well, welcome back then. And today I really wanted to focus in on the Bee Lab. It's a global leader in bee biodiversity data, and it has been a cornerstone of the field for decades. I mean, I think when I got started, it was already, moving and, was an inspiration. So if you could take us, let's trace the origin. Where did it get started? What were the circumstances that led its focus to bees, and what are some of the key milestones that shaped the lab into what it is today?
I'll truncate a lot. So I've been working almost 50 years here at Patuxent, and my theme has been design, development, and running of different kinds of monitoring programs for different groups. Originally birds, and then it evolved as I went to a now defunct agency called the National Biological Survey. those were the scientists from the Department of Interior Agency. So I was Fish and Wildlife originally, moved to this NBS group. It blew up, as things do politically. And instead of putting us back, most of us went into the US Geological Survey. So I'm now here with about what was formerly about 2,000 positions. We're way down the list. And so I came back, but with a broadened horizon of the groups and the creatures that we could look at. And a lot of what we were doing at NBS was like, okay, what can we collect information for that will prevent future train wrecks, as the wording was, and species endangerment? And so we went through phases. So we did a whole thing on amphibians, and a lot of those programs are still lurking around. And then looked at the next things. I played around with, like, what about mushrooms or fungal communities? Too difficult. The variance, you have to be there at only certain times of year, and it was not tractable, but very interesting. Then it was pollinate. Well, calling crickets and katydids, right? So insects that are announcing themselves. How unusual is that? That was pretty good, but I couldn't get anyone really interested other than me and maybe 5 people. And also you had to have really damn good hearing, because a lot of their songs are right at the edge of our hearing. My hearing was deteriorating at that point. So drop that one. Then it was like, okay, pollinators, because the book Forgotten Pollinators had come out, and I started poking around. I got connected to the Logan Bee Lab, and just as you do when you're starting something, asked around and asked a lot of questions, and went out there for the first maybe 7 or 8 years. I would go every year to Logan Bee Lab to sort of drink from the fountain of native bee information, and would stay with Frank Parker, and worked with Vince Tepedino and several other people. So truncating that, we started working on, as a side project to some of the others, trying these techniques ourselves. I should say ourselves. I always use the plural when most of the time is just me going out, putting out different bowl traps, netting, like, how does this work? Am I getting a lot of things? And so that became more and more of interest. I had a butterfly phase in there before this, because I knew how to identify butterflies, and I read books and things, and then realized that North American Butterfly Association was starting at that point. I talked to Jeff Goldberg, I think, or I could have that wrong. And so we would go back and forth, and he had a little bit different vision, but it was also a vision of coming up with information about status of butterflies. And I was like, these guys are going to do it. It may not be the way I would do it, but let's have them run that. And then I turned my attention to bees, which I knew essentially nothing about other than general entomology stuff. And I was like, I can't identify these things, but I can run the traps. We hired Harold Eichert, who now works and has worked for years at the Logan Bee Lab, to come in and do identifications. I actually had money back then. And so we started playing with the survey techniques and things, with an idea that we would move this into some kind of national monitoring program like we had done for other sorts of things. So that never happened. And the reasons were a series of investigations into the sampling variance of survey techniques for bees, which are horrendous. We have a statistical pass. And so a lot of what you need to know in terms of like empirical data about monitoring something is you need to have some sort of estimate of year-to-year variation in your proposed technique. And that will tell you most things. And then you can divide it by the mean. And we've done this we'd have a bioscience paper with James Gibbs. And what we found is, not super surprising, plants, very low variances, right? They don't move. You can go back. There they are. Elephants, big things, low variances. You can find them. Then you go into the insect world, huge variances. When we looked at the data, we're not talking the goal under 50%, 20% would be great. We were talking average 150%. It just the math didn't work. So we shifted to primarily being a support laboratory, developing techniques, and it became clear almost immediately that identifying what you've captured is the bottleneck. it's actually relatively easy, particularly with traps, to catch bees. And you could catch millions of bees. And we never really started big volunteer programs because of that, because we couldn't handle the volume. It was me and volunteers, and then me. And then 5 years ago, we hired Sydney Schumar. And then Clare joined our group. She's going to talk about it, different aspect, Fish and Wildlife Service. But the bottom line was we couldn't make it work at the scale it needed to work. But we realized we can help other groups do a better job of what they have the resources to do. So we focused on how do you catch bees, evaluating betterness, which is there's lots of different metrics to why you would use one thing versus another, putting together the handy bee manual, or officially the very handy bee manual, and the Discover Life. We started working with them to develop in a very collaborative way again too, having different groups put things together and developed online ID guides, because again, the bottleneck, what am I looking at here? And in the old days, you had this much literature. You can't even see my hands. There's so much that you had to have, and it was revisions, name changes, like, oh, these two are actually the same. There's one article on that. It was not tractable. So we try to distill a lot of that into Discover Life, and it's an ongoing thing. So we developed that as support. Our photography came along with that to act as a online museum, and we keep expanding those facets. So those are the main features, our criteria. We're not part of a museum. In fact, I never got permission to start the program. So this is for you young people, don't ask permission, because they would have said no. But over time, they were like, oh, I guess we have this bee program. And then we would give it different names as we went along. So maybe that's bad advice. But in that time period, we were able to expand mostly because we lived in the interstitial spaces of USGS science and they weren't paying attention. And then at some point, they liked us. So we had on the books this year a $35 million new pollinator research center and all kinds of staff coming in. That's on hold. We'll see what happens. And at this point, that's sort of where we're landed, and we'll follow up with that. I want to emphasize we're not a museum, so we don't have a collection. We have reference material, and then we have a huge backlog, which equals probably 100,000 specimens, because we have a huge backlog. We're committed to, if we catch something, to pinning up everything and ultimately identifying it or shipping it off to someone who will do that. And then we maintain on the back end a database. I kind of taking up a lot of room here. So I'm gonna shift to Clare so she can talk about the walk of Fish and Wildlife Service to get to the same point. But those are some of the, painted with a broad brush, how we got here.
Thanks,
Now I
Clare.
can
Yeah,
have some
you have a different trajectory, but you're in the same location. Can you describe your path as well?
Well, I have a little less time in the biz, so it's a simpler story. Yeah, I am with the U.S. Fish and Wildlife Service. I came in through a fellowship program in the late 20-teens, and I live locally, and it was a pollinator fellowship project, and Sam is right down the street from my house, relatively. And, I got to benefit from the generosity that he's built up. He helped me identify my specimens and checked my things. And by the time that project was over, I was deeply in love and very stubborn. I would only take a job that had a microscope with it. And it was the kind of right time, right place. The Fish and Wildlife Service was getting really, really ramped up in their pollinator work and dedicated, funding and positions for that. So, like I said, I really wanted a microscope job, and I told everyone about it. And right place, right time, and they were able to bring me on for that. It was a good pitch too, that they were already paying me through contracts to do the same thing. So, they were getting the work done, and out of convenience and making sense, we landed here. There's not really a local place that this kind of work could have been done anyway with the Fish and Wildlife Services office locally. So this was just the space that made sense, and we're all friends. Yeah, so that was in 2021. I started with science applications, and then in 2023 moved over to the refuge system. And at that point, I also received a contractor who is now half of my brain. I really, really appreciate that extra support. And we are also support staff for Fish and Wildlife Service, primarily refuge biologists, but clearly we don't isolate ourselves in that respect. And we just recently put out a framework for an updated framework for the Fish and Wildlife Service, how we want to inventory monitor bees, the whole slew of refreshed protocols, and all of these. I'm sure we'll talk a little bit later about the special issue in the Journal of Melitology that came out with the Research Coordination Network. But those papers came out, and we just really worked hard to make everything cross-compatible. And the other kind of landmark thing that we do is some of the data management. So Sam referenced the database that we have on the back end, and considering I was on some of the standardization papers, it just made sense for us to be able to be a good example of adhering to those. So for the past year, working really hard on getting another data set up to GBIF, and we just did that. So that feels like a real win. And those pretty much cover, and we do some training activities as well. So those kind of cover our main points.
Well, congratulations on getting the data, getting that data loaded up. I know it's a huge endeavor to make sure everything is correct before it goes up. And the thing I love about this story is, I was thinking, I heard Bob Pyle recently speak, and just the way in which like the Xerces Society came together, it was kind of a gamble, and he'd been in Europe and he saw something, and he didn't really know where it was going to lead. It's quite remarkable. And I was thinking about the same thing when you were peering down the rabbit hole, Sam. You probably didn't, you ran into some obstacles, but they have generated a number of resources that all of us use now. That in some ways, some of these journeys don't have you don't know where they end.
Right.
You just know that something needs to get done, and then you'll work it out later. And if you thought it out and put it on paper and created the blueprint, it may never happen.
Yeah. I'll mention that this is why you want public servants, right? None of the stuff that we're doing would get you a professorship or would get you tenure or even would make a great, certainly not a PhD, maybe a master's, I don't know. And then Claire and I share lots of resources. We're literally in the same building. We share the reference material. We equally work on kind of keeping the lab up to date. You can't do something, I can do that. It's a really nice collaborative space to work in. And then we have lots of visitors. So another big facet of what we do is, particularly east of the Mississippi, but we leak over sometimes into the Midwest, is you're the poor grad students, okay? I'm really interested in bees, so I'm going to do this project, and I collect all these bees, which you need to do because of that variance. And then you're pinning it up and like, okay, now I'm just going to identify them, then I'm going to analyze my data. Then you start with the first one and you spend 2 hours on the identification of one, and you have collected 15,000 specimens. So we help out, and it can look a lot of different ways. So with some particular things, we will actually process and pin and label and use our database for the system. In other circumstances, they do whatever they do. Hopefully they're talking to us before. That's what I want to mention. We're collaborative, but it pays attention to talk to us it's the best, I should say, to talk to us at the beginning of your project. So we can say, well, you could do that. We did that. It really was a nightmare. We have made all the mistakes theoretically that anyone could make. That's what we're good for. Maybe not that, maybe this, or, that kind of considerations. And then we can help you with the workflow. So we're really good because we process thousands and thousands of specimens every year with, okay, here's many, many small considerations to think about. And then in the end, we would like you to try to identify your specimens, because that's something that we need more of those kinds of people. And it's helpful to us to have something sorted, even if it's there's a lot of gaps and uncorrected things. It's like more like I'm sure the Oregon Bee Atlas is doing a lot of these kinds of things from what I know, so that in the end, Link doesn't have just this hodgepodge that he has to go through. The fewer things, because we're dealing with thousands, we have to pick up and move, the better. And they learn some things. And then we are acting as, in a way, somewhere between a mentor and almost a contractor, although we don't accept money, and they're the apprentice. So we want to apprentice that, okay, we'll go through these, we'll make the corrections, we'll enter them if it's on our labels, or we'll give them back to you and you enter them. And that is almost required, because nobody can just pick up, say, I'm interested in bees, I'm going to start a project, and then using existing tools come up with the correct IDs. You're always working with people, but that's not obvious to many people in the field who are just starting, or you have a major professor or something who says, I think we can do a pollinator project. But that's not like doing a bird project where most of the parameters. With starting a native bee project, you don't realize a long string of things. A lot of that's in the handy bee manual. There's a lot that you can do with the online material, but really you want to develop a partnership. It doesn't have to be us. There are other bee heads out there. A partnership with someone who has some kind of on the ground experience, so that you maximize your work and don't go down places that don't yield as much. So anyway, again, we feel like we do these things as public servants, and that's a very useful thing for us to do. But we ended up never starting a monitoring program. There's several in the works. Oregon Bee Atlas can be seen as a monitoring program or an inventory. That's great. Let us support you in ways by talking to us. So similar things happen all over the place, but a big national program like we've done in the past for birds and amphibians, nope, not us, not now. Takes too much. It's a resource problem. It's not something that can't be done. It's something that can't be done with the resources available. And then there's philosophical questions about, well, what is it that you're trying to address with a national program? If one was to start, are you trying to address changes in, your basically crow and sparrow bees? That would be interesting. But what about all those rare conservation bees, the ones that probably do need some more focus? And thinking about all those things, including just the impossibility of really tracking in a statistical way, or a strong statistical way, a lot of rare species, to looking at the relationship of plants and bees, because the solution is almost always going to be plant-based, whether it's saving the sand dunes and maintaining the specialist plants, or it's just making sure that we retain and integrate more native plants of certain kinds back into systems. When you plant a pollinator field or a CRP land, what do you plant? So these are more what we're trying to tackle now, mostly through gathering standardized information about bees that use plants and visit flowers at what rate, and that allows comparisons versus the old school, which is useful, which is, I caught this bee on goldenrod, right? But how many other plants were there that you didn't get them on, and what goldenrod species, and all these questions? All you have is that singleton thing. But it's like, if you have a lot of goldenrod, eventually everything will be on goldenrod. And that's clearly, there's more to it than that, right? There's things that only go to goldenrod, and things that are like, yeah, I'll have a sip of goldenrod nectar. And then similarly, if you don't look at a particular plant, then it looks like that specialist bee, willow, phacelia, whatever it is, has disappeared. And so more in that direction, to target vulnerable, rare, and specific relationships, and then bringing that back in a variety of different ways, working with people mostly. That's what we mostly do, is collaborate. Bringing that back to the guy on the mower, right? When do you mow? How do you mow? do we herbicide? Do I plant a pollinator meadow here? Or do I work with the woods and try and bring in the vernal flowers that are the part of the East? Or do I plant a bunch of Rudbeckia and Echinacea in an eastern pollinator garden when those two species are essentially waifs and rare? You see what I'm saying? So
Yeah.
all kinds of interesting things from a conservation point of view that don't necessarily require us to do a monitoring program, but is helpful. And developing the techniques to do that.
Yeah, that's really helpful. And I guess you know, there is this impulse to create like a, something like eBird or something that will monitor and be able to track them, all the birds. But we have this problem, as you've pointed out, of, in technical terms, I've always heard it referred to as spatial temporal turnover, that there's, as you go through the same landscape this year and next year, you may not see the same things. If you go a week before and a week later, it changes. If you go 10 miles down the road, there's a real issue associated with this. And I love the idea of kind of pivoting from that into collaborations that might be able to shed light on some of these more specific questions as opposed to this blanket question. And it would be, give us a snapshot of some of these initiatives. So you talked about the grad student and you've, laid down the warning before you start your project. Talk to maybe somebody who has a background in bee taxonomy or natural history to sort of come up with a sampling guideline and just, before you spend all that time in the field. What other kinds of collaborations, and I imagine apart from like the graduate student, are you involved with?
Well, we do a lot of giving people specimens. So we have in the database, I don't know, Claire, what, 200, 800,000, 200, whatever that number, very large number.
And I think
we're
we
not a
just
museum.
posted a little over 500,000.
Yeah. And
we have, that gives context of how many we are still backlogged on, but over 500,000 was in that last push up to GBIF.
Right. And because we're not a museum or a monitoring program, and we do a lot of collaborative work, a lot of times we're working with someone. Like this year, we have several collaborations going on. In the end, it's like, here is your specimens back. And so they leak away. And even if they don't, we just don't need 1 million of each species, even if there could be, there's always something interesting in them. But again, we're not this program that is tracking change over time. So we have less of an onus to keep large amounts. So we're very good. Like you're interested in a particular taxonomic problem, we'll give that to you. So we work with a lot of molecular people at a lot of different levels, and we're very happy to give away specimens. So we do that all the time. That's one type of collaboration. So another type of collaboration is the collaboration of, you need your specimens identified. So we have worked in every state east of the Mississippi and most of the Midwestern states directly, like getting specimens from them or us visiting their museums, because I like to visit museums and look at their specimens, because I'll be like, oh, I have not been able to see that species before. Now I can score it more completely in the Discover Life Guide, which is an ongoing thing. Other collaboratives are, like we work with people at the Logan Lab, mostly grad students, to develop Western field guides. Like we will help you, and this is a pitch to all your listeners. You want to expand any of the Discover Life guides to the West? Come talk to us. It's not rocket science. It's really using the literature and using your eyeballs. So you don't have to have a PhD in taxonomy, but we're not going to be the ones to do it. We really don't have a reference place that's Western. We really stop in the first tier of states, east, west of the Mississippi, in terms of our understanding. And there's too many. So, but we have a number of guides that go all the way to the West. If you see things that you'd like to change, talk to us. It's easy to change. We can give you permissions to make those changes, pictures, additional information, and so forth, that would make the Discover Life guides useful to a particular area. So we work with people all the time on that in terms of incorporating them into doing different facets, loading up the literature. is a good one. So a good place to find obscure literature is to see, so every species of bee has a species page, which is maps, pictures, and literature primarily. And so, but if you don't load the literature, you don't get it. And so having people load literature is one of our favorite little collaboratives. But we work a lot with National Park Service. Clare is Fish and Wildlife Service, so she's the go-to person for a lot of projects there. And lots of academics. So again, in the East, if you look, our intensity of collaboration is really tight around the lab. So all the universities in Maryland and DC, we're working with their students and doing things hands-on, sometimes in the field with them. And then it spaces out from there. And a lot of times, the further away you go, the less they have a personal relationship. So we give lots of talks. And another sort of, I guess, weird collaborative space. So you would never do this. I'm not going to say never, but if you're primarily a academic or a government researcher, research grade, you have measures that say, we, the Committee of Higher Learning, whatever, are looking for high value publications and certain kinds of things. They're not looking for giving a talk to the garden club. So we feel that kind of outreach, which we do regularly, respreads that messaging back out. Another sort of collaborative thing is I like going out with people and seeing their particular bee area and learning from them directly. I'm very much a boots on the ground. This morning I was out, I live on the backside of the Patuxent River, so I was down in the swamps putting out new traps. And we haven't talked much about it, but another big facet is development of survey techniques. So recent ones have been an expanded trapping technique for emergence from nesting sites. So you might know about like the bug dorm and the kinds of approaches. Well, they're just like a meter by a meter, and it costs unbelievable amounts of money. So we're working and developed a technique that allows you to run a infinitely large array of netting over sites and catch the bees that are emerging. So because it's very difficult to find bee nests, that's one problem that we've tackled recently. And now USDA, we work with them on that, and they're going to publish it, which is another good thing about the Bee Lab. You want to publish this, we start things, you finish them, we're looking at something new because we don't get any credit. And there's a political end to this where I just have stopped being an author on journal articles for my own reasons. This year is the year of using brine in traps. So, a lot of traps are like colored something, main trap, cup, bowl, filled with soapy water. You put it out, starts evaporating immediately. And you come back that day or the next day. And then if you want something that lasts longer, use propylene glycol, very expensive, $30 a gallon, and also a mess to clean afterwards. So what we're working on now is something I was aware of, but I never put it together, which is replacing the soapy water with a completely saturated salt solution. That sounds fancy, but it's a cup of salt and a gallon of water, and you pickle anything that ends up in there. And it's got a couple advantages. One, decreases evaporation rate by 50%. Two, nothing goes bad. They're pickles. So you can keep adding water to the cups, and now your trap window expands way out. We have processed specimens from vein traps where they just use, this is us doing it too, after 3 weeks in July. Ooh, it works, but it is really bad smelling, and a lot of the specimens are really on the edge. But with salt, not a problem at all. So anyway, we're trying in different places. We're also trying to develop and have developed a hanging trap that is a stadium cup, like a 12-ounce stadium cup, which is just a stiffer beer cup that you would buy at Walmart. And just using some, off-the-shelf kind of wire, fill that with soapy water, hang it from blooming shrubs, and we have to go through the whole process to ID these things, but it looks like it might be a really good way to get an understanding of the bees going to particular woody plants.
Huh.
You can use an expanding pole and hang them from willows and things like that. And the problem is almost all the blooming woody plants all bloom at the same time. And so you have that small window, good day, middle of the day. I'm one place, one person with a net. You can only do so much. But if you have traps, you can put them out whenever, let them run the whole cycle of the plant's bloom cycle and retrieve them. And you have more information.
We were just telling our volunteers that, we have, disproportionately few records on maple. And we tell them, but you know what, you got to get the low hanging branches. But whenever you go to the low hanging branches, you can see all the activities way up high. It's like, can't get it.
Well, this is a good technique to think about. So first, buy one of those 3-level telescoping poles for paint or for pool cleaning. And then take a bicycle rack screw, so it's a bar with a tip up and a screw at the other end so that you screw it into the wall, hang your bike. Okay.
Oh, yeah. Yeah.
Run that into the tip of your pole, and now you hook that onto the wires of your cup, not the hook, but the wires, and you lift it up, drop it wherever you want on that tree, pretty high. I'll do it aside. I spent a lot of time gazing at flowers, and this spring I spent a lot of time gazing at maples. So Eastern observation, they're supposed to be good for maples. Maple's supposed to be good for bees. We have red maple, silver maple, Norway maple introduced, and a couple others. The native ones are not that great. They get some syrphids, a little bit of action, but Japanese maples, Norway maple, unbelievable in terms of the amount of activity on those things. Red maple, silver maple, sugar maple, kind of like nothing.
Huh.
Anyway, that's part of what we do. These natural history things over time sink in. I put in the traps in those. This is why I'm particularly gazing at them. Like, I'm going to see what's in those maples. And so we hung a bunch up there and got really almost very little. Now, if I put them in a chokeberry or a rosaceous plant or a willow, thing loads up. Anyway, I'm down on maples, Andy. Cut them down. Bowling alleys. That's all they're good for.
Well, thanks for that sage advice there, Sam. And Claire, just on this idea of collaboration, there is the refuge system across the United States, and there has been, for all sorts of taxa, just an ongoing monitoring project. In some ways, I imagine this is part of the mission of these refuges. Can you talk a little bit about the collaborations, the kind of collaborative work with the refuges?
Yeah, I would be happy to. So each region does get to, make their own decisions, obviously, and I work on a headquarters level in that support capacity. And so it depends, but often it's kind of similar. A refuge or that region, like with the Oregon Bee Atlas, will collaborate with somebody outside, typically a university. But I think we have others that are, maybe working with another federal or state agencies are usually our other prime collaborators. And my part in that, right, the pollinator inventory monitoring team of refuges is, like Sam said, it's really good if you call us before. And at least in the few years that I've been here, that word has gotten out, and I think we get pretty good contact before and planning. And then what they are focusing on sometimes also brings in different partners. So for example, we have a project in the Southeast that is, instead of just doing, broadcast bowl cups or something like that to fill in data gaps, they are using their capacity to focus on specific focal areas that are of interest to the refuge system or that refuge. And they're going around the region every year to different places. And so that partnership happens. Sometimes we use ACE, American Conservation Experience interns, and then some of those might stay on as volunteers or contractors or things like that. And they might also be working with the partners program, which depending on region is either sitting in refuges or sitting in ecological services, but that's, some inside baseball there. And then I also serve on teams like the Native Bee RCN or AFWA, Association for Fish and Wildlife Agencies, and things like that. But those end up being more of this kind of collaborative, I know the word is collaborative, but it's less about the physical surveys and more about how are we getting all of us sort of on the same page so that all of these choices, right? Each project makes different choices based on their objective, but at least we're all collecting in the same ways and we can compare our data, or they all go up into occurrence maps and we know how it was done. But I also will give a little bit of a pitch there. We've said a couple of times there's challenges with having literally people to get identification. Sam was mentioning that we really like things to come to us already sorted in some skilled way. And I admire the master melitologist program so much. I really want one on the East Coast. I hope it happens at some point. But we've been having good success with these refuge projects is having those interns, those techs that are on them who, often are early graduates of either undergrad or grad, or maybe they're finishing up a capstone project. They will do a work study here. So they might come, we had just had a really good group come in the spring to learn some of their field techniques, and they'll come back in the fall to learn how to process and get to genus level with their IDs. And we've had really good success. So I'm now getting a couple of projects that are pre-sorted in some way. And in fact, two young women who did this in my first year of trying to run a work study now have full-time positions at Fish and Wildlife Service.
Oh,
And
wow.
they do identifications and they send them here and we use the data and it's really, really helpful, especially like you said, they're catching, I think, over 15,000 bees a year. It's really helpful, and I'm excited about these ways of kind of collaborating in a not necessarily a completely project hard science-based way, but more in a development way.
I'd really like to pick up on this theme. So I think for many people in the West, we know the lab. There's a big footprint of the lab in the West, even though many of your support roles are in the East. Just in terms of the trainings and Discover Life and the very handy bee manual, sorry, that there's a real huge contribution that has been made from the lab to build up capacity. And it does resonate with what Sam said earlier, that when in the early days of contemplating this whole endeavor, that it was clear that a handful of taxonomists would not be sufficient to be able to resolve some of these sticky questions around bee conservation. Maybe for people who don't know about these resources, you've touched a little bit on them, but maybe just talk a little bit about the suite of them, the kind of resources that are available nationally that are helping everybody. We do have an assignment, I will say, in the Master Melitologist that involves going through the Discover Life citations and then going back to the original source. It's really, it's so well organized. It's fantastic.
Let me punt to Claire, because Claire and her technician Sally have been really spending a lot of time putting together a much more professional looking set of materials, and they have I don't know, Google Drive, but I'm gonna let her explain that and also the taxonomy.
Happy to. And thank you for mentioning Sally, because that was the first thing I was going to give big props to. So like I said, in 2023, I was fortunate enough to be able to bring in a contractor, and now she's functionally my lab manager, and she's the one creating all of our media products. So a few years back, we updated the Very Handy Bee manual. I think we're going to get another one out in the next, I don't know, don't quote me on that, year or so, because yeah, she's made some incredible training products, the different videos for even like Sam going out and netting bees. And so everybody can get that wisdom virtually. to different kind of like one-pagers for managers and for students and things like that. So I will give credit where credit is due. Any beautiful digital media comes from Sally Len. Call her, she's great. But you were asking more also about the identification guides and those kinds of trainings. I'll also give huge credit to Rob Jean, who I took my genus level course with, because just like Sam, this was a kind of DIY teach as we go, teach ourselves as we go. Lucky to have reference collections to check our work. And so I kind of take the kind of the schedule that he offered us and some of the, key lessons, the spot it, you got it's, and really try to focus on those when I'm doing work study with the students, because usually where they're at is they're going to be right field technicians who we honestly really want to make sure that they know the diversity of bees, so that they catch, they know that a nomada is a nomada and not a wasp, and then they go look for it. So that's an objective of kind of prepping people for identification before really going down the microscope, end of things. So we kind of have that spectrum. And in the fall, they would come back and I would kind of copycat on Rob and some of the lessons he taught me. And then, and that has worked really well. I really enjoy doing that. Sam and I might try to expand that out to more workshops, in-person workshop or something like that. But speaking to the BID classes, so again, since 2021, 2022, Sam received a microscope with a camera so we can live stream bee identification. And I love this, and it's incredibly gratifying when folks reach out and say that they're using videos. So I think we have over 160 something videos. They're recorded, they're posted. I'm sure I can share that for show notes and things like that. We did have some IT problems last year with some of the changes. So those are up now. They were in it for a little while. And the first 30 or so are all genus level characteristics. And, as we're moving through, Sam might flip back and forth between different cheat sheets that he might have made or other people have made. Other people's keys, we'll introduce other keys. I'm pretty sure that we demonstrated the Pacific Northwest genus guide at one point. We had link visits. So I think that was probably on that episode. And then also the original literature will sometimes be flipping back and forth through all of those. So I think those classes are really helpful for introducing people to a whole suite of resources, not just relying on Discover Life, or not just relying on a dichotomous key that if you get one thing wrong, it really sends you down the wrong path. And one of the things that I think that those classes are really useful for, besides just we're going through us at this point, we're really in the weeds on some Andrina, and we go through 2-3 specimens a class. But especially in those early days, it helped folks kind of know where they can jump ahead in a key. Oh, I know that group of Andrina. I kind of get that subgenus. I can skip to where the dark scopa are, that kind of. And you don't, even in classes, people, you might take an identification course, or I don't know, a taxonomic course. These are kind of the more, just kind of the inside, here, let's get this a little bit quicker. Here are some tips and tricks that folks seem to appreciate. And I find it incredibly gratifying when, particularly grad students, clearly there's a need there for grad student education support in their universities on these skills. They tell me that they've gone back through the recordings, or they ask for different specimens or species. So that's super gratifying. And out of that have come a few enhancements to Discover Life, which I think I'm going to turn over to Sam in a second to describe. And we have a wonderful friend, David Kappert, who takes incredible photographs. And you haven't had him on here, I suggest. And he had a huge effort in making a great photographic bee glossary, and also guide to Andrina specifically, to kind of help break down these barriers of, oh my God, what's a tegula? Okay, here's the tegula. It's a little wing covering thing, so folks who don't necessarily have the background in the jargon, we can still figure it out, right? You don't need to know all of the official terms to start looking. You can figure it out as you go along, learning it in layman's terms and then kind of backtracking. So yeah, I think that's what I have to say about that. And I'm going to turn it over to Sam to describe the Discover Life keys.
Yeah, let me just point out a couple things. One, there is no need for any glossary to be terms other than David Cappert's. It's that good. Second, Clare has somewhere a secret page filled with all the links and tables and things that we
Right.
use, including links to Dave Cappert's. So a lot of this is not, Google it and it shows up kind of stuff. So I would suggest that maybe Claire sends that to you, Anthony, you post it with this thing.
That's a good reminder. I will say that class, the bee identification, BID weekly sessions, we do have a partner, like a Google Drive that goes with that to share out.
Also, everything we do is public domain. So anything you see, you can use in any way you want, like our photos, the Discover Life keys. You can copy all the keys and make them your own. We would prefer that you just work with us and we'll make them better. And again, we're happy to collaborate with anyone on expanding these, but you can just take everything.
Two things on that. One, with the classes, we also do have often cameos from the people who wrote the papers. So sometimes that is another thing that helps build the guides. We had a real long session with Joel Gardner and Jason Gibbs on the dialectus that we want to kind of return to. Mike Arducer was on a lot when we did a Bumblebee series. We had all the hot names. It was a lot of fun. That was a good series. And so that's another way that we're building and making sure that those papers are being shared, right? Sometimes if you're not keeping up on literature, and in the literature sense, in this Google Drive that I'll share, it has all the literature that I'm aware of so far. And I have a great library, like access to library through the Fish and Wildlife Service. So that's another resource if folks need keys, guides, if there's something that they can't access. We usually put them in there for papers, new pubs. Those all land in there when it's at the, with the will of the people, the will of the class.
I'll also point out that we are not stuck in the East. So if anyone listening wants to do a session with Claire to talk about bees of, the upper Pacific Northwest or whatever, that's great. We also have done at least a couple sessions that are the natural history, the bees of this area, and talk about them and their rarity and their conservation too. So the series is available to lots of different people, and over time it'll become more and more findable, one hopes. And so that's another opportunity. Dialing back, there was a mention of the difficulty sometimes of picking out these parasitic species. So particularly with people just starting. So training is good, but we also tell people to just collect all the waspy looking things and bee looking things. And then when processing out specimens, particularly from bowl traps or vein traps or things like that, we tell people pin up all the wasps because a lot of them aren't, we can use pretty relatively low level people, and we can also do double checks with the bycatch that gets set aside, keep them here, we'll look through them. But the nice thing is that we have them pin up all the wasps and bees. We don't ask them to try to separate them. And then when we go through, we set them aside. And for years, we've been sending them up to Alberta to Matthias Buck. And he identifies them, sent us back a spreadsheet. And so we now have actually great wasp data. And also we have circumvented the problem of people overlooking Stelis and Nomada and Sacodes and Hylaeus even as not a bee. So anyway, just so.
Oh, that's great. We have a lot of wasp bycatch that's already databased.
Mm-hmm.
All right, cool.
Anyway, I can put you in touch with Matthias and see, probably more interested in your Western stuff anyway.
Okay, well, so this is great, and it really sets the stage. I think, coming from the starting point, it's clear that a lot of capacity has been built through this initiative. And maybe just zooming out on the big picture, from all of these efforts that you're, all these ways that you're contributing and sort of facilitating, how is this work translating into conservation for native bees in the United States? Are there some examples of where you can point to and show like this is starting to have or has already had long-term impacts in our capacities?
Yeah, it's a little bit to disentangle our efforts from the larger desire to push pollinator things. So I think we facilitate that, right? So if you don't go out and collect bees or look at bees or count bees or start a bee project, then those contributions are nil. And so if we didn't exist and didn't help people, we know that, for example, a lot fewer projects would get done. Or the information, not that we're perfect, but that the information on the identifications would be a lower quality. And that usually means that the rare stuff sort of disappears. And so one of the big factors is quite a number of a trail of papers that are first record for this, first record of that, or interesting observation or those kinds of things. And you can see our fingerprints on that because we did the identifications and maybe said, hey, this is a really interesting story. A lot of the introduced species that have come into North America in the East have our names on them because a bunch of people like to do urban bee and suburban thing and or are in the kind of disturbed habitats to agricultural where the invasives like to show up. And so again, we're there to say, this doesn't look right. And so we have, I can't remember how many, but a bunch of the first specimens of things showed up in our collections or we identified there. It's a little bit ineffable because of that. If you look at acknowledgements, I can't remember, someone, maybe Sydney, who's the lab manager for USGS in our powerhouse of two people, she, I think, did a search of acknowledgements, and like we were mentioned just the bee lab for 800 times in the literature. Wow. I guess the way to think about this is that our goal is not to get our name on things, right? We don't need to be publishing. In fact, we don't publish. We don't need even the acknowledgements. There's no money transacting here. We're not building up our business. Our prestige is self-determined by how well people like our material and whether they use it. So we have a very different MO than most people. So it's a little bit difficult to wrap the minds around because we're not angling for anything except to help you. That makes me really happy. That's why I'm a public servant. I really love this work because I get to help people, and that's my job, is to help people out. I learn things all the time. I'm a lifelong learner. I'm out hunting bees all the time. I then get to distill this and put this into practice in a public-facing way. We also have, we didn't mention this, we have now with Hollis Woodard as a co-conspirator, a listserv of about 1,400 people that people can join. They can either contact me or Hollis or find it. It's a Google Groups called Bee Monitoring, which focuses on monitoring types of things, but basically it's natural history to monitoring things. And there's a very nice archive that you can search. So you want to, what's the best way to use a vacuum, bug sucker? What's the latest? I can't find nets anymore. Those kinds of questions, great for the listserv. We encourage that. So that's another tool that's in our hip pocket. So it's that kind of stuff. I can't really go like, look, we won all these awards or great, but there's a lot of the mycelia. There's a lot of mycelia, not a lot of mushrooms. How about that?
Thanks for that, Sam. So, Claire, how do you see this issue of how all of this work and the ways in which it's really had this large, very broad effect, having conservation outcomes?
Right. So, from the Fish and Wildlife Service perspective, Sam spoke to like having fingerprints on everything. We're not quite so prolific in the literature yet, though I know that there's plenty of folks that have been deeply involved. I think you got Joe Engler out there, who's ex-fish. And what we are trying to do is use all that information, all of this like historical, wow, there's 20 years of a lot of data in certain places, and use that to make decisions on our refuges about where we're going to pursue survey efforts. Instead of putting out, massive full track collections on places that like in regions that either have a lot of data already, or, we kind of understand the habitat that's kind of in, like, maybe let's think about something else. So some of our projects are approaching it from we have these focal area habitats, or we have this T&E species. Let's go there and make sure that we are filling in a really needed gap. That also, because these are on refuges and some of these are fairly remote, it's not something that will likely easily get picked up by, a student or community science without, a lot of effort. And so some of those are designed to just really literally get a baseline of what's in this wetland. Nobody's looked at a wetland in this state. Maybe we should try to a couple other projects that are, and we're talking a lot about adaptive and informed management, not perfectly statistical before and after models, but there are definitely other refuges that are attempting or trying to plan for a pre and post survey to the best of, the capacity, or going on a place that's being treated and another site on their refuge a distance away, and trying to do some comparisons to make some like sense, some like gut check, some preliminary information that they can use to start making decisions, because as we know, it's going to take a long time to get really narrow information back. But if we're condensing, refining, making more precise decisions about where we're going to be collecting, we'll be able to turn that back faster as well. So that's kind of where we're going. I don't have a lot to say, oh, this has been the conservation outcome. Here's a really great case study for it. But those are the ways that we're approaching the conservation of the wild bees that we do have to collect to understand.
I really love that answer because it's tackling this really large problem, the rabbit hole that Sam was peering down so many years ago, from two angles, I guess, creating the scientific basis for understanding and making patterns. But then things like refuges are ongoing, it's not a blank slate that you start from. It's an ongoing management project where you will want to try something. And if you have the capacity now to look at pre and post, it can give you some sense of, yes, do more of this, or this was a lot of money and it didn't have much of an impact. It really starts to fill in the gaps for making, conservation that's not shotgun. It's much more focused and directed.
Yeah, for sure. And I think we're trying to also think about it of, you collect and you go searching and you find those specialists, or you don't. And maybe that can be, a real early indicator of, hey, this is doing well. This is not doing quite as well. Maybe look at your plant community. Do you have a lot of maple trees there? Maybe that's a good thing. That's not a good thing. Do you have a lot of loosestrife? Maybe that's a good thing. That's not a good thing. So those are kind of how those things are being used.
This is some of the tricky area that we see a lot, which makes sense from people's point of view, coming from often a wildlife management perspective. I'm interested in deer. I'm interested in birds. I want to measure things. I'm doing a treatment. I want to see the impact. I'm putting a pollinator garden, or I did something, and I want to see an impact. It turns out that from a bee point of view, that's really tricky to measure and be conclusive about it. You can catch a lot of things, and that's why, from my point of view, but everyone has their own agenda, so it's really difficult to really move that ship. Just start gathering information about what bees you have. That's a really good tractable thing. Don't try and add these layers like, oh, we're going to measure vegetation with it. Well, your ability to measure vegetation is also not that great, and two not greats do not make a great. Or oh gosh, well, we just put in all these plots that are new forest things. It would be great to have see what the impact of adding those was, or whatever. Well, the problem is that you need lots of replicates, lots of time, and lots of specimens, and it still will be fuzzy. But doing those kind of surveys that are more what I call farmer science, where you go in. So like a farmer will go like, gosh, I think I'm going to plant soybeans early this year. And then, oh my God, they did not grow well. I'm not going to do that again. So you just have to go out there and catch things and look at things. We don't have enough of that base natural history information that provides insight into what should be doing. People are applying honeybee stuff. They're applying deer management, bird things, vegetation, and like mixing it up. Like Claire said, many of these refuges have 0 to a couple INAT, bumblebee on their list of things. These are the places that need to be done. A lot of these refuges have opportunities. So do DoD sites in particular, because DoD sites are like the worst habitat to farm. So they're filled with sand. They're filled with all kinds of edaphic conditions that are uncommon. They have all kinds of rare bees, but we don't know that. And then what do you do with this information, is another thing to think about a lot. So you have a correlation or no correlation with vegetation. Well, what are you going to do with that, right? Where does that add? What body does collecting more things on more plants and spending all the time doing that versus understanding that, oh, we have a set of bees that are globally uncommon. Let's focus in on that. It seems like one of the problems is you're making something very complex that should be really simple. Just finding stuff, finding what's uncommon. Where is it? And then extrapolating back, like, oh, we have this rare Hylaeus, but it likes, and we know something about it. We like sand, but we're planning to, plant that all with pine. Maybe then the conclusion might be good for everybody. Maybe not do that. Anyway, I could go on and on about these kinds of things, but our ability to measure bees and impacts of bees and bee populations is low at the detail. The big picture is more records of uncommon things and associated natural history. iNaturalist is a great generator of many of those things. The Oregon Bee Atlas is a great example of things that have direct application. It's like, oh, I don't know what it would be. We have these osmia that we didn't know we had that are on mints in the left hand of the Blue Mountains. And now we can tell people, hey, plant more of that, or don't mess it up, or whatever. So I think a simpler ideal of what we're doing, which is looking for stuff and trying to find it, and then applying our conservation to some of those findings for these uncommon species. I would be super happy with that.
It's really interesting. I was thinking about how the podcast started, where, looking down the rabbit hole, not quite realizing how many, I don't know, dens and turns there would be. It seems like at this point in the lab's history, it's many of the kind of obstacles are been identified, or they're, it's a little bit of more of a, maybe it's still intractable, but it's a tractable problem now. Like there's certain avenues that you've talked about. And so I guess what's next on the horizon? What's the future of the Bee Lab and this endeavor, and where does it go from here?
Yeah, so I think Claire and I have different tracks and we have same tracks. So we're both committed to the joint building, and the reference collection is really a unique piece of technical equipment in a way that we have to have, because it's old school. Like you go through identification, then you're like, this is a rare one or a tricky one. I'm going to go look at and do this. Oh yeah, they're the same. Or no, not something's off. And it's still at that level. So that's our shared future, is working on that together. We have, as everyone knows, there's administrative issues that are difficult to discern right now. We're here. I find that to be a small miracle, because our budget, not the Bee Lab, no one cares about the Bee Lab, but at the scale of the ecosystems mission area, which is about 2,000 people, has been 0 from the president's point of view. a reflection of Project 2025. So I expected that we would be gone because it says 0. There's a bunch of smaller issues there, but we're still here, and we're having a difficult time moving forward with large projects or big expansions or visions of the future. The whole $35 million bee, native bee thing is on hold, all that. We're still doing good stuff because we've come from a background where our budget was $3,000 a year. So we're still getting paid. It works. We have lots of volunteers. We're collaborative. So that's good. I'd say intellectually, in terms of future things, we're always going to be working on techniques. So it's just shocking how, well, what about this? What about these different colors? What about fluorescent colors? What about different detergents? What about, different configurations? It seemingly is endless. So we have all kinds of students that we work on there. So technique development, ID guides with Discover Life, always going to need tweaking. We're always a little bit behind the taxonomy. We are always trying to work with volunteers to make that better. Yeah, like I'm saying, we would love to work with West Coast people to bump out some of these guides or make changes that they're based on them actually looking at specimens, types of things. So we see the bulk of what the USGS side does continuing. Our ambitions are way scaled back in terms of trying to do national work. So we're working with those groups like Neil Cobb and Hollis Woodard's projects, and there are the Xerxes Society, a bunch of these folks. We were just at meetings to work on conservative values for bee species and applying those. So we're volunteering our expertise, but we're not looking for money and things like that. So that's the kind of thing. We'll be lucky if we are still here in 4 years. We'll consider that to be a success because of the targeted nature of our larger group. The signs are on paper negative, but the fact that we have seen no clear actions is the positive part. And so I don't know what that means, really. It just means that we're still here and we're still doing good things, and don't expect any grand new projects, but we'll help you with your grand new project. How about that?
Well, I certainly speak for all the audience that of the show, and also people who work in bees. We hope that these investments are made because of the outsized effect that this initiative has had nationally in bee conservation and just understanding, what's going on with bees at a fundamental level.
Yeah, I was so elevated by the outpouring of support that came without any real campaign, because we can't really do that. But we had Xerxes and a bunches of other groups writing to their congressional representatives with their feelings about these things. And it was huge. And that just made me feel the love. And I really felt like the things that we had helped people out in the past had grown, and this was the return of that fruit. So I was really gladdened by all that. Thank you, everybody. And all the many different people had played roles, including the people that helped Claire and myself move out of our old lab building, which still exists, but mostly for wet work, and move into a new building that used to raise endangered cranes, but chicks. And now we have our glass top drawers, and we feel like we could walk away for quite a period of time without the mice and the dermestids eating everything up. So yeah, good.
Well, feel the love, you guys. Thank you for all that you do.
Thank you, Anthony. Thank you for having us.
Thank you.
Thank you so much for listening. Show notes with links from each episode are available at the website pollinationpodcast.oregonstate.edu. I also love hearing from you, and there's a form at the website where you can pop in and say hello and give me feedback. If you want to support the show, remember to leave a rating on iTunes, Spotify, or whatever podcast mothership you use. And finally, if you have the means and you want to help support my lab's effort to document bee biodiversity in Oregon, visit OregonBeeAtlas.org and follow down to the donate button where you can make a tax-deductible donation to the Jerry and Judith Paul Native Pollinator Endowment. Every little bit helps. See you next episode.